From Microbe TV, this is Office Hours for Wednesday, January 10th, 2024. Got to get used to saying that. I'm Vincent Racaniello, and welcome to your little viral corner of the internet. It's good to see everybody here again. And let me welcome our moderators first and foremost. I see Les, I see Steph SF, and I see Frank and Vanity Nutrition. Welcome, everyone. And Tom Steinberg as well. Thanks, everyone. Thanks, moderators, for coming. And thanks to all of you for coming back. I always wonder when no one's going to show up. And I can't do the stream with no one. I need at least one person. At least one. I'll do it for one person, but not for zero. It's like a class. If one person showed up, I would teach it. So you may notice I'm wearing a Got Polio t-shirt over my regular shirt. This was a holiday present from Karen. Thanks, Karen. Got Polio. Me neither. Thanks. How do I do the fingers? Thanks, science. Yeah. And then it says, or diphtheria, pertussis, tetanus, measles, mumps, rubella, HPV, Haemophilus, pneumococcus, hepatitis. What else is there? Hepatitis A, hepatitis B, and more, right? A lot more. They couldn't put them all on the shirt. Yeah, vaccines work. And they don't kill you. No, sorry. Sorry, all of you who think they kill you. They don't. They protect you. If you get a vaccine and you get in a car accident and get killed 2 weeks later, it's not because of the vaccine, but that goes on the vaccine adverse events site. Anyway, to pull off it, and I talked about that today. So you get to hear it next Monday when it drops. And I'm wearing the polio shirt also because I want to give you guys a little polio mini lecture today. All right? I'm going to do that when I'm alone. No guest tonight. Mini lecture. Next week we do have a guest. But I will wait to announce it because we don't have all of you here tonight. But let's find out first. Let's see, what else do I have to do? I now have show notes for Office Hours to tell me what to say. Okay. I have a story to tell, but I want to find out first who's here. I know that Les is here from California and Lise is here from Ohio. I know that Abdul is in Saudi Arabia. Ooh, look. Characters that I cannot read nor pronounce from Singapore. That's great. You know, I might be in Singapore next year. Who knows? I'm going to ostensibly twice to Australia. And you know, it's only 400 hours north of Australia. So I could go to Singapore. All right. Sorry, I just ate. I got to do that. Singapore. Who else do we have here? Rima. Hello, Rima from Iowa. I know John is from Minnesota. Tom is from Eugene, Oregon tonight. Vanity's from Long Island. Steph is from San Francisco. Nicola is from Italy. You know, MN said earlier, he said, he or she, I don't know. Where is it? Come on, Vincent. Why can't you find it? Anyway, MN said, every time I hear his name, I think, here you go. Robert Bedoin said, Bodwin. Every time I hear his last name, it reminds me of that song Black and Yellow by Wiz Khalifa. Yeah, that's how my students remember me. One year they said, Professor, whenever we hear that song, we think of you. Thank you so much. Heart. You can do heart thingy without the thumb. It's a little harder. I'm not good. Thank you. Yeah, my students. And I tell people, it's just like Black and Yellow. And if they're of a certain age, they don't get it. But young people do. Yep. Philip is in Wales, UK. Abhilash is in Frankfurt, Germany, that I passed through a few weeks ago, last year in December, actually. Who else told us? Changsha, China. Will, welcome back. Christella is from Ohio. Frank is from here in the New York area. Kang is from Chicago. -1 and cloudy. What is it here? Let's see. Put up the weather here in New York. It is. Come on, bit 6 degrees and windy. We had a massive rain and windstorm last night. Yeah. MK is from Eastern Massachusetts. Visto tutti. It's like, oh, visto tutti. I saw everything. That's a good name. Sydney. Hello, Rima. We miss you. I'm here every week. Try the veal. Hey, if you're on Instagram, you get a preview of the comedy, the stand-up I did at the meeting, the giant virus meeting. I'll put the whole thing on YouTube soon. That was cool. In Saudi Arabia, it's 50 Fahrenheit. Yeah. What does the shirt say? I believe I read it. Got polio? Me neither. Thanks, science. Okay. It's gray. It's good. It goes with my black shirt. Watertown, Wisconsin. Claire's from the UK. Here's Andrew, our other moderator from Pongaroa. Welcome. Oh, look who's here, everyone. Welcome Amy, the virologist. There she is. Welcome, Amy. Thanks for weighing in. And Amy's here to fact check me tonight. So I'm gonna give a little polio mini lesson. And many people have seen Amy already. Where can we buy this shirt? I don't know. I mean, she bought it somewhere. Karen, if you're listening, where do you buy the shirt? I haven't seen her, so she may not be here. I'll find out. You like this shirt? Zan is from Australia. Well, you have a question. I will get to that. Simon is from San Jose. Doreen loves the mini lessons. Thank you. I'm glad you like them. You never know. Mike and Lori are in Seattle. Doreen is Hartford. Maureen is in Northeast Ohio. Elizabeth, West Virginia. MN, near D.C. Tim is in Toronto. Ooh, Dr. Nair is in Malaysia. Wow. That's pretty far away. Steve and Lori are from Paso Robles, which makes wonderful red wine, as you, of course, know. Cheryl is from Santa Rosa. Jem is from Hampton. Are you a Jem? I bet you are. Catherine is from Peru. Wow. Cool. We had someone from Colombia 2 times ago, I believe. Now we have Peru. Lovely. Here we have someone else from New Zealand. Otepoti. Logic is from the UK. CW is from Virginia Beach. Robert is from Louisiana. Bodouin, I guess that's where the name comes from. Very cool. Hans is from Ecuador. Oh, look at that. We have 2 South American representations. I bet we're going to have more. Carol, our favorite nephrologist from Southern California. I don't want to hear what the temperature is. Sorry. Dean is from Wisconsin. Peter is from Boulder. Look at this. Where to buy the shirt? I could sell merch on this show. People love it. It's great. You know, folks, if you want your T-shirt sold here, send me one. I'll wear it. But you can't, it's got to be a science-related one, you know? You can't put other stuff on. Eric is from Ohio. Yeah, we got that. Come on down to Australia. We'll have the prawns ready on the Barbie. What does the Barbie mean? A barbecue. Oh, yes. Yeah, I'm going to go 2 different places. Let's see. Where am I going first? Cairns. Notice I said it right. Cairns, and also, I think Brisbane. I think I'm going to both. And 2 separate trips. So, would be lovely to see some people. Lori is from Tomball, Texas. Ruth is from California. Andrew is from, and another Australian. Good day. Baton Rouge, Louisiana. Amy, you see people are very happy to see you. Austin, Texas. Central Connecticut. Ontario. Lehigh Valley, PA. You share my birthday. I've watched weeks of you and consider your family. Thank you. Thank you so much. Let me try again the love sign without the I'm not very good at it. You know, the women do it better because they have long fingers. So I just use my thumbs. Sorry. It's not meant to insult long-fingered people, but I have short fingers. I'm not very good at it. Anyway, thank you, EHS, for listening. Yeah, happy birthday. We just had a birthday. You're probably much younger than me. Jessica's in Toledo, Ohio. Salt Lake City, Tom, who ran into Nels last week, I understand. Jane is from Michigan. Lori, you heard about British Columbia. Ohio's in the house, you bet. So Simon is from the UK. You live in San Jose. Well, we're interested in where you are now. Moisha is from New Orleans. Greg is from Seattle. Is there a date set for the first lecture of the 2024 course to drop on YouTube? I just try and get them up as soon as possible. So the first lecture is a Wednesday, which is basically next Wednesday, a week from today. Yeah, I'll be busy. And I'll try and drop it by Friday. We got a busy production schedule, yeah. Let's see, Seattle I already checked in. Jeff is in San Diego. And Levis is from Colombia. Hola, Levis. There we go. We have Colombia, Ecuador, and Peru. Our representation from South America. I can't speak Spanish. Okay. And Amy says, we're starting to work on the 8th edition of Fields Virology. Amy and I co-authored the last, was it coronaviruses? Yeah, chapter, and we're going to do it the same. It's our encore. All right, so I want to tell you a story. This is very funny. So as you know, I was in Germany for the giant virus meeting in December, early December. And, excuse me, sorry about that. Matthias Fischer was my host. He told me a story. He said, Matthias Fischer discovered Ma virus, which is so named because it's related to a transposable element called maverick. So he called it Ma virus. He said he got an email from a guy last year who said, there's a card in this card game called Mavirus. I don't know how to pronounce it. So Matthias sent him an audio file. And it's on YouTube. You can find it. Here's the link. It's called Netrunner, How to Pronounce Ma Virus. And this guy is a player. So these are the cards. Netrunner is a card game, kind of like Magic cards, right? And here's the Ma Virus card. And he didn't know how to pronounce it. So he made this video saying, well, I wrote the guy who discovered it, and he told me how to pronounce it. He sent an audio file. And so it's Ma Virus. The funny thing is that Neon Static mispronounced he mispronounced Matthias's name. So I wrote in the comment, when I visited Matthias in Heidelberg, he told me this story, showed me the video. Also, his name is pronounced Matthias. Matthias, not Matthias, Matthias. Haha. And he said, sounds like I need to add a new video to the series. So that was pretty funny. But I didn't even put the screen share on. I'm really sorry. I guess you didn't see any of that. Darn. Anyway, so this is the card game. And that's Ma virus in the story I told you. And then down here in the comments, I wrote a comment and he responded. And I told him how to pronounce Matthias. So by the way, it is very funny. At the meeting, Matthias was recalling this story to a few people, including Curtis Suttle, in whose lab Matthias discovered Ma virus. And Curtis said to him, "That's not how to pronounce it. It's Ma virus." And Matthias said, "No, it's Ma virus." And Curtis said, "No." And Matthias said, "I discovered it. I get to name it, and I get the name how to pronounce it." It was a pretty funny exchange. So anyway, go listen to that video. I'll show it to you again. Just go. I searched for Netrunner Ma virus, right? You'll find it. Okay, so that's that. It's a funny story. Well, maybe I already told you. I don't know. Could be. Sometimes I repeat things. You know how you know about that? You know how that goes? Let's see if I have all the I don't have all the names up here. Microbe TV / contribute. Yeah, that's another one we should put up here. We always love to have your support for what we do, of course. And Daniel Griffin during the month of January is doing a matching donations at parasiteswithoutborders.com. So if you go to that website, yeah, it's like Doctors Without Borders, but parasites really have no borders. Neither do viruses or bacteria or air. So many things don't have borders. Can you list all the things that don't have borders? Anyway, if you go there and you donate, he will double your donation. He will match it. So that's a good place to go until the end of January. And then after that, go to microbe.tv/contribute. You guys are very generous with your support. With your support, we built the incubator. We do all these programs, these science programs. And we tell you what's real science. We don't tell you fake science. So if you want real science, come here. All right, let me give you a little polio mini lecture, all right? Why am I doing this? Well, some people seem to think we can eradicate poliovirus or even polio, the disease. Now, there's a difference, right? There's the virus and there's this disease it causes. So I want to point out the problems with that. But I want to go back, to the very beginning and tell you a little bit about how we found poliovirus. So first of all, poliovirus is an acute infection. Now, last time we talked about varicella zoster virus, or herpes virus, that causes a persistent infection. In other words, it lasts the lifetime of the host. Acute infections are circumscribed in their length. Not necessarily short, but they can be. Polio, rhino, rotavirus, influenza viruses, they come and they go within a certain period of time. You either die or you recover. So polio is an acute infection. This is the virus, a small particle, 30 nanometers in diameter. You can see the very thin protein shell. This is a cutaway, which shows the RNA inside of it. Now, poliomyelitis has been around for thousands of years. We have this Egyptian carving, which shows probably a record of a priest with what looks like polio, drop foot, because the muscle is paralyzed. You have flaccid paralysis. You can't move your muscle, so you can't use it, so it atrophies. But it didn't become epidemic until around 1900. You can see the number of cases versus the year. Suddenly, in the 1900s, we have hundreds, thousands, and then tens of thousands of cases. And so the first question is, why did this epidemiology change all of a sudden? Well, it turns out it is a disease of modern sanitation. Before 1900, polio circulated freely, infected most babies shortly after birth. Feces were everywhere. There were no toilets, no sewers. You used a chamber pot in your room at night, right? And then in the morning, you threw it out on the street. Great practice, folks, yeah. Anyway, polio was everywhere because it's excreted in the feces, and so babies were infected very shortly after birth, but they had antibodies from their mother, and that prevented them from getting paralysis. So in 1900, what happened? Dixon de Pommier calls it the greatest invention of humanity, the sewer and the toilet. Yeah, and now we delay infection. Babies are no longer getting infected shortly after birth. They're getting infected when they're 5, 6, 7, 8 years old. And now there are thousands, tens of thousands of kids who are now susceptible. They don't have antibodies. And there you get your outbreaks of polio, epidemic poliomyelitis. It wasn't a mutation. No, it was not. And of course, there were mutations happening all the time in the genome, but that's not what caused this change in epidemiology. It was a change in sanitation. Okay, now poliomyelitis, humans are the only known reservoir. It's spread by fecal-oral transmission. What does that mean? The virus is ingested. You have some fecal contamination on your hands. You don't wash your hands after you go to the bathroom. You shake someone's hand. Kids don't wash their hands. We have terrible hand hygiene. So you eat some poliovirus, it goes into your intestine, reproduces extensively, and it's shed in the feces to infect someone else. In the intestine, it crosses into the blood, establishes a viremia, and in about 1% of infections, it gets into the central nervous system and causes paralysis. Only 1 in 100 kids on average get paralyzed. All right, we have 3 serotypes of poliovirus, pretty much a human-specific virus. And in the 30s and 40s and 50s, hospitals were full of iron lungs like this to help you breathe. You would be paralyzed in the respiratory muscles, so you were put into these negative pressure chambers to help you breathe, and eventually most of these people recovered chest muscle function, and they could go on with their lives. Now, you may know that FDR, president of the U.S., had polio. He could not walk on his own. He had leg braces. He used a wheelchair. So he raised money by virtue of the National Foundation for Infantile Paralysis and the March of Dimes. And that foundation paid for the research that led to the 2 polio vaccines we use today. And I want to talk a little bit about those. Now, here's the curve of polio past 1900 in the U.S. So here, 1900, big outbreak in 1916, and then increasing numbers fueled by the post-World War II baby boomers, right? All these new kids, me included, I'm a baby boomer, 1953. They provided more substrate for infection. And then in 1955 and in 1961, new polio vaccines were introduced and have drastically reduced the number of cases here in the U.S. and globally, as you'll see. So today we have 2 polio vaccines. We have the inactivated vaccine, which is what we now use in the U.S. It's injected, doesn't produce intestinal immunity. It's injected into your muscle, but gives you humoral immunity to prevent CNS invasion. We used this vaccine from 55 to 61. It was then replaced with the oral polio vaccine. It was still available, but it wasn't recommended. We switched to the OPV in 61. This is in the form of drops that you ingest and replicates in your intestine and produces immunity. And these are basically attenuated viruses produced by selection. They're mutants. In 2000, we went back to IPV, and this is currently what we use in the U.S., and we'll talk a little bit about why that is in a moment. So here are the effects of the vaccine. The inactivated vaccine introduced in 55 brings down the cases dramatically to a few thousand a year, and then the oral vaccine eventually leads to removal of wild-type virus-caused disease in the U.S. I don't say wild-type virus because we haven't done enough environmental surveillance to know that. There was no more wild-type polio in the U.S. after 1979, although you'll see there has been another kind of polio. The inactivated vaccine and the oral vaccine induce antibodies in the blood that block invasion of the CNS. And initially, the oral polio vaccine will immunize the gut, but only for a short period of time. Okay, so in 1988, the WHO said, we have great polio vaccines. They said, let's try and eradicate poliomyelitis by the year 2000. This is the original document published by the 41st World Health Assembly, May 13th, 1988. Look at the name, Global Eradication of Poliomyelitis. Poliomyelitis, okay? But over the years, it became Stop Poliovirus Transmission. Feature creep. I don't know how that happened. I don't know who couldn't figure out the difference between poliomyelitis and poliovirus. Suddenly it became poliovirus, which is impossible. You can't eradicate poliovirus. Maybe you can eradicate poliomyelitis. So what happened? 1988, I don't know, 350,000 global cases of polio. The countries in red have polio. Then 10 years later, a lot less. 10 years later, even less. We had just Nigeria, Pakistan, India, Afghanistan. And today it's even better. But I want to first show you the problem with OPV, oral polio vaccine, which is being used for this eradication campaign. In the U.S., here's reported cases of polio from 61 to 2003. Here's total paralytic polio in the line, cases on the y-axis, year on the x-axis. In 1961, we switched to the OPV vaccine from IPV, and suddenly we see cases of polio, these gray bars, that are caused by the vaccine, so-called vaccine-associated paralytic polio. After 1979, which was the last case of wild polio, that is caused by circulating wild poliovirus, all the cases of polio were caused by the vaccine. And that's why in 2000 we switched back to IPV, which does not cause polio. All right, so in the U.S. we do not use OPV. It's been delicensed. It causes polio. We don't want to do that to our kids. However, WHO is using OPV in its global campaign. So where are we? These are the number of cases as of January 2nd, my birthday. Global wild polio 1 and circulating vaccine-derived poliovirus cases. So what do we have here? First of all, wild polio 1 still circulates in a number of countries shown on this map. We have Afghanistan and Pakistan, those largely because of immunization issues. But then we have a lot of circulating vaccine-derived polioviruses, which, as I said, they're because the vaccine can cause polio to a certain extent. And why is that? Because it reverts when it replicates in the intestine. All these green dots in Africa are caused by vaccine type 2 OPV. And there are also cases elsewhere. You see there was a New York case caused by OPV2 in 2022, and there have been cases in other countries, which aren't shown on this map, by the way. This is a map from polioeradication.org, which just focuses on Africa for some reason. There are cases elsewhere, folks. And these orange are vaccine-derived type 1 cases. All right, so in 2016, so far, types 2 and 3 have been declared eradicated, at least disease caused by types 2 and 3. And it was noticed that a lot of the cases of type 2 were vaccine-derived. So in 2016, they took out OPV2 from the vaccine. Big mistake. So here are the isolates of OPV2 after polio 2 after OPV2 withdrawal in 2016. They're AFP specimens from people and their sewage samples. And you can see 2016, 2017, really not seeing any type 2 poliovirus. The problem was that there was undetected type 2 in Nigeria, this province over here, Borno State in the northeast. Type 2 had been undetected but circulating, so they resumed monovalent OPV2 campaigns to eliminate the circulation, and that has reintroduced type 2 into the world, basically, vaccine-derived type 2. So you can see here, here's 2016, right? Very little type 2. And then when you go back to eliminate that Nigeria outbreak, you see more and more type 2. And that's where we are today. We have a lot of circulating vaccine-derived polio 2. All right, so what do you do about this? First of all, we had a case in New York caused by circulating vaccine-derived OPV2, probably brought in the U.S. by someone who had been immunized. The problem here, wastewater testing in the New York area showed that there is poliovirus circulating in people, and therefore it's in the sewage. And most people are immunized, so they don't get polio. This is as of February 2023. The reason there was a case in Rockland County is that immunization rates had dropped way below the national level, and so this individual hadn't been vaccinated. All right, so what do you do? This type 2 OPV is circulating globally. Well, you can't use IPV because it's still going to circulate. So a number of investigators, including people you have heard about on TWIV, Andrew McAdam, Raul Andino, and also Kostya Chumakov, re-engineered the type 2 OPV. They introduced a number of mutations in the genome, which I won't go into, which were thought to prevent reversion and causing paralysis. And this is called NOPV2. It was authorized by WHO, given to millions of kids in various African countries. No reversion in the 5' end, which is what happens typically with OPV2. But unfortunately, subsequently, a number of kids in DRC and Burundi got paralyzed after being given NOPV2. And these viruses are recombinants in which all of the genetic modifications that were introduced into NOPV2 were removed by recombination with enteroviruses that we have in our intestines. So NOPV2 is not the answer. It still causes polio. It's about 5 times better than OPV2 in terms of causing polio, but it causes polio and it spreads to other countries. So what do we do? First of all, forget about poliovirus, OPV, as a solution, because we're full of enteroviruses that recombine, and no matter what you do, you can try and modify till the end of the world. Those viruses will recombine with our enteroviruses in the gut. So forget that. But really, the question is, can we eradicate a disease? We eradicated smallpox in 1978. Here are the requirements for eradication. Replication in one host, so polio ticks that box. Vaccination must induce lifelong immunity. Probably polio ticks that box. Asymptomatic infections don't contribute to spread. No. 99% of polio infections are not paralytic, let's say. And so with that, you cannot eradicate. With smallpox, every infection gave you symptoms. So even if we switch to IPV, which globally not likely we can do, these OPV viruses that we have there will continue to circulate, and we don't do very much wastewater testing, so we don't know what's out there. So we need new polio vaccines. We could use IPV, but injection globally, probably not going to happen. We need a vaccine that induces mucosal immunity. It's not infectious, so it can't cause polio. However, here's the catch. Research on polio is being terminated by bureaucrats who think polio research is not needed. They think the vaccines we have are fine. Well, I'll tell you, folks, paralyzing kids is not fine. Not in my book, anyway. And if some countries stop immunizing at some point because they think the disease is eradicated, we're going to have outbreaks that rival those of the pre-vaccine era, which would be tragic as they're totally preventable. So we cannot eradicate poliovirus. We'll never have sufficient global surveillance. Maybe we can eliminate the disease, but we can't eliminate it by using a vaccine that causes the disease. All right? And finally, let me just close with this letter to Science, written by Stanley Plotkin and Kostya Chumakov. Polio eradication efforts, above all, do no harm. Although vaccine campaigns that use the improved vaccine, that's NOPV2, would prevent polio caused by wild virus, they would still cause cases of polio. This plan is unacceptable when a safer, more effective vaccine protocol exists. Unacceptable. Yes, it's unethical to give kids a vaccine that is not licensed in the U.S. OPV, or its derivatives thereof, an NOPV. It's not ethical to give them a vaccine that will paralyze them. Why is it good enough for kids in Africa but not in the U.S.? So what are we going to do? We got a vaccine that works, IPV, but are you going to inject it into everyone globally? I think that's hard to do. We should make a new one. So you should be allowed to do polio research. People are saying, in a couple of years, we're going to shut down all polio research. Have you thought about it? Have you really thought about doing that? Because there's poliovirus everywhere. What's the danger of doing research on it? Oh, my God. I've worked on polio for 40 years. I've thought about it a lot. And I think the people involved in this have not thought about it for 10 minutes. Otherwise, they wouldn't do this. So that's why I wanted to talk about polio tonight, because crazy things are happening. And, you know, the Gates has poured billions of dollars into the eradication campaign. And that's lovely. They've eliminated a lot of cases of polio. But has anyone told Mr. Gates that we can't eradicate poliovirus and maybe we can't even do poliomyelitis with the current vaccine? Has anyone had the courage to tell him? I bet not. What would he say? You told me. He also paid for NOPV2. Let's answer some questions, folks. Sorry to rant for half an hour, but I'm very passionate about that, as you may know, because I worked on it for so long. Okay, here we go. Farhad, during your past work in the lab, was there any procedure or particular virus that made you uncomfortable to perform a test on? So I worked on poliovirus mainly, and a few other enteroviruses. I was never uncomfortable working with any of them. We also did a lot of mouse work. We paralyzed a lot of mice. We injected mice with viruses to raise antibodies. And I have to say, I didn't feel good about doing that. All right. I know you need to do animal research. I felt badly, right? For many reasons I don't want to go into. That was the one thing that made me uncomfortable to do. Yeah. Me personally. I'm not talking for anybody else in my laboratory. If someone had hepatitis vaccine many years ago but don't know which type, can they have a new vaccine again? Sure. Yeah, just get them both. It doesn't hurt. No, you can get. Well, there's only two, right? Hep A and Hep B vaccines. Just get them both. Better be protected, right? Let's scroll down here and see. Paxlovid in Mexico would probably cost the equivalent to several months of minimum wage. So that sucks, right? It shouldn't be. We're a global community. Yeah, sure, Vincent. Be optimistic. I know what you're saying. We're all humans in this together. Why can't we provide medicines to people who can't buy them? Countries like the U.S. could do that, right? We make expensive weapons of war, and a fraction of that could take care of medicines for everybody. Why? Do we not care about other people? Is that the problem? Well, that's it, yeah, because otherwise we would be helping people, right? So Zan is in Australia, flying to the U.S. to see mom. Got newest COVID and flu vaccines, RSV is not approved. Okay, so what's the question? You're going to get infected, but you won't get severely ill with RSV. Yeah, if you're not vaccinated, you could. I don't know how old you are, but, if you're young and very old, that could be a problem. But if you're in between, don't worry about it. Okay, here we go. So, Vincent, after what we've learned, don't you think we need to phase out all attenuated vaccines and replace them with alternatives? Well, I would say no, not a blanket commentary. I think, measles, mumps, rubella are very good. They're stable, attenuated vaccines. They don't have the problems of polio vaccines, which revert and recombine. They don't do that. So I don't think so. I don't think we need to replace all of them, no. What are the differences between these three serotypes of polio that all three are required to cover against paralysis? Well, they all look like the poliovirus that I showed you earlier. There are subtle differences in the surface which make it so that antibodies against one of the serotypes aren't as good at protecting you against disease with the other two serotypes, right? They all cause poliomyelitis. They have slightly different properties, but antigenically, which means the surface proteins are slightly different so that we needed to put all 3 in the vaccine. And you see, when we withdrew 1, it's a big problem. We withdrew type 2, and now we're paying for it ever since. I have been reading A and C's chapter. A and V, I guess, right? Is there much of an update? Oh, yeah, we're going to majorly revise that chapter. It'll be driven by Dr. Rosenfeld, for sure. What makes it emerging? I think the 8th edition, if I'm not mistaken, is no longer going to be called emerging viruses, but certainly polio and other entros are emerging, right? Other entros recombined with polio, you get new viruses made, and other entros are changing in their epidemiology, much like poliovirus changed at the beginning of the 1900s. So, yeah, remember, emerging virus is either a new virus or one whose epidemiology is changing. And that's certainly the case for the enteroviruses. New study in cell host and microbe Alessandro Sette. Memory, CD4 and CD8 T cells continue to provide cell-mediated immune recognition to highly mutated emerging variants such as BA2.86. Yes, that's because the variants change in their proteins that are recognized by antibodies, but they change far less in the proteins that are recognized by T cells. And so T cells, of course, they provide help to B cells to make antibodies, but they also will eliminate virus-infected cells. So, the antibodies can't prevent infection with a new variant, but once the infection gets going, your T cells come in and they eliminate the infected cells, and that's why you get infected, you have symptoms, but you don't develop severe disease unless you are over, say, 70 and have other comorbidities, in which case you need to have a plan to take an antiviral, for example, and hopefully in your country you can get it, and I'm sorry if you can't. If you make me the world's health czar, I will assure that everyone gets what they need. But you need to give me money, all right? You need to give me a lot of money and step back, leave me alone, and I'll solve it. But I don't want to do that, actually. I want to stay here at Microbe TV and do science communication. Okie dokie. Hello, Kip and Laura. Thank you for your contribution to science communication. Vaccines give us humans the once unheard of power to choose health. What a good statement. I love it. The once unheard of power to choose health. Yeah, you can choose to be healthy or not. It's up to you. Here we go. He changed it to A and V's chapter. Yeah. Same question, but corrected. A and V. Yep. Vincent, I was in China over Christmas. About 1 out of 5 people masking. No one panicking about any virus surges, despite all the news claiming otherwise. The news has but one goal: to get you excited so that you will read their news or watch their news. That is their only goal. They are less interested in telling you what actually is happening. Can you imagine a headline? The new variant does not pose any particular problem. They would never, ever publish a headline like that. It doesn't get them any viewers. That doesn't get them clicks. No page views. So they say the new variant is going to kill everyone. SARS-CoV-2 is changing regularly antigenically. The outbreaks are not caused by the changes. They are caused by people changing their behavior. Think of polio emerging around 1900. Not blaming mutations, but rather human behavior. The invention of a toilet for that. Here, let's see, in December, what are people doing that there are more infections? Is it a new variant? No, they're getting together for the holidays. They're traveling. Kids are coming back from school. Could we think about this, news outlets, and not blame the variants? You know, Amy and I wrote an op-ed a couple of years ago in the New York Times. Blame human behavior, not the variants. People don't care. It's not sensational enough. So don't tell me the variant is causing a problem, because you don't know that. It's human behavior mostly. Okay. I seem to be on fire tonight. What science, if any, do you, I like that, if any, do you use to try and increase the reach and spread of your science shows? Viewer numbers aren't matching the Nutter conspiracy shows. Can science help? Oh. Yeah, we don't get the views that the other people do. All the people spewing nonsense, spewing anti-science, they get far more views than we do. But we're getting more and more. So there are two things that I'm depending on. We're just going to keep getting more and more people listening. Many of you have told us that you tell your friends what we say on our programs and they tell their friends. So there's this long tail effect without people actually listening. But I'm also hoping to inspire other like-minded scientists to start talking about their work and start talking about science, okay? And together I think we can grow this. We're trying to use social media more and more. So we have Kim at the incubator now. If you're on Instagram following us, Microbe TV, you will notice that there are bunches of new videos coming out regularly, which I wasn't able to do before. I just didn't have the bandwidth. But we'll record things at the incubator. She'll take snips out of shows that are particularly noteworthy, and that attracts more people because they're short, right? I don't know, even me doing stand-up comedy could attract a person or two. So our strategy is to try and do that. We do long programs, right? 1 to 2 hours of discussions that are heavy. That's not going to do it. But that attracted our core audience. And from that, I think we can grow. That is our philosophy or plan. I heard today in British Columbia, they are starting a home swab kit for HPV screening. Interesting. All right. So that's great. What is this? Who's the audience for this? Okay, if someone's 50 years old and they test positive, it's too late to do anything. It doesn't have an impact on cancer rates to immunize them at that age. So who's going to be swabbed? A kid, maybe. I don't know what age people become sexually active. I have no idea, but I'm guessing it's in like the teens, maybe early to mid-teens. So your kid becomes a teenager, swab them and test them. If they're still negative, vaccinate them. If they're positive, vaccinate them too. It can still have an impact. So I'm not sure what the purpose of this kit is. I have to read it, of course, but I'm just giving you my thoughts here, off the cuff. COVID symptoms are changing to sore throat and congestion. Is this due to evolving virus or host or both? It's due to immunity. People are immune. They got multiple vaccines. They get infected over and over. It's immunity that is making the infections mild. Virus is not changing. There's no reason for a virus to become milder. People talk about this all the time. I used to talk about it. No reason for it to become milder. It doesn't make any sense because even the most lethal virus, Ebola virus, you're still able to transmit it before you die. So that's nonsense. So it's all immunity, I'm convinced. The UK Office for National Statistics found that JN.1 is causing insomnia and anxiety as symptoms. Yeah, they're reading the frickin' news and they're getting anxious. Thank you, news outlets, for making people anxious. No, it's not the virus. It's the news reporting around it. What biological process might cause that? Yeah, these are neurological processes. Oh, there's the major causation would be when you get an infection, you make cytokines. You have an immune response, and those can get into your brain, and they can have effects. There's no question about that. So it's plausible that they could cause insomnia and anxiety. You know, it would have to be a side effect of the inflammatory process. I don't know why that would be different for JN1. There's just nothing that makes sense there. I think the press is making them anxious. Good job, folks. Be nice to your neighbor. Throughout the history of science, there have been institutions propagating dogma and silencing what came to be known as true. Do you consider yourself and our institution to be pro-free speech? Well, this probably got moderated, I would guess. I am totally pro-free speech. I'm not censoring people who talk about fake science. I'm not saying we should shut them up. I just think you shouldn't listen to them because they're wrong. But if you want to listen to nonsense and waste brain power on it, you're welcome to. But then you should listen to us and find out why they're wrong. I'm not going to speak for institutions because I don't really, that's not my thing. But I can tell you that I don't silence people, but I don't bring them on my shows because they're not telling the truth, and I don't want them. Yeah, and Simon got it right. Caused by people having been told too many times to worry about variants escaping immunity. Thank you, Simon. We think similarly. And you asked this question last week, but yeah, I don't think I answered it. Right. How about that for memory, huh? At my age, I can remember. You asked this last week. Do you think science would have been so successful with COVID if Musk had taken over Twitter when the pandemic began? Yeah, I would have been, because the people making those vaccines could give a rat's ass about Twitter, X. I mean, there would be less dialogue, but that was not essential for the creation of vaccines and antivirals. So I'm not going to give Musk any credit for either doing good or bad. Is anyone eligible for Paxlovid? Anyone 12 or older with mild to moderate disease at high risk for severe disease. All right. Now, physicians, I know I hear this all the time in the emails to Daniel. Physicians refuse to give it to some people. You don't need it. You're not a candidate. It's going to make it worse. It doesn't work. It causes too many side effects. Come on, folks. Daniel Griffin every week tells you you're wrong, you're wrong, you're wrong about all that stuff. Why don't you get with it and give people the drug they need? You know, most of the people who are dying don't get Paxlovid, and if they did or they had, probably would have saved their lives. So you're hearing lots of people not getting them because the doctors choose not to give it to them, but they should. They are eligible. Some coronaviruses are neurotropic. Is SARS-CoV-2 a neurotropic virus? So, to quote my colleague from Field's Virology, I don't know if we have it in there actually, Dr. Rosenfeld, neurotropism is the capacity of a virus to reproduce in neural cells, in your brain cells, in your spinal cord, in your peripheral ganglia, because they have neurons in there too, right? No, SARS-CoV-2 is not neurotropic. I have never seen any good evidence. Now, you can grow nerve cells in culture and infect them. I don't care. I want to know if you ever get replication in the brain, and I haven't seen any good evidence that suggests that. It is not neurotropic. Dr. R, would you agree with me? will you be attending the annual Virology meeting 2024? Where? There are lots of them. I think, if I remember, you're from Peru. I'm not going there, no. But if you want me to, if you invite me, I'll go. Sure. I'd love to go to Peru. I hear it's beautiful. But I'm going to the annual meeting of the American Society for Virology, which is in Columbus, Ohio, this year. Yeah. How does antibody enhancement work? Like in the dengue second infection. So, you get infected with dengue. There are 4 serotypes. And if you get infected with serotype 1, you make antibodies and T cells against that serotype. Those will not protect you from disease caused by the other 3 serotypes. So let's say you get infected with type 1 dengue, and then a year later you get infected with type 2 dengue. Your memory B cells are going to make antibodies to type 1 because that's what they saw before. The type 1 will bind the type 2 virus, but it will not block its infection, and it will allow that virus to now get into cells that normally would not be infected, like macrophages that have receptors for the antibody. So the virus now, in the presence of that non-neutralizing antibody, can now infect new cells, make more virus, lots of cytokines, and that's where you get this more severe disease. Okay? There you go. Mm-hmm. I've decided to be annoyed that it's shortened to polio because it's from poliomyelitis, which is from Greek, polios gray and myelus marrow. Yeah, it's in my lecture, of course. You don't like poliovirus either. It used to be called poliomyelitis virus for many years, and then it got shortened. You know, the nomenclature people took over. And so, yeah, polio is just gray. You're absolutely right about that. Neil Young's song Helpless was about his polio hospitalization. That's right. Helpless, helpless, helpless. Interesting. Yes, war is good for poliovirus. War is good for infectious disease. I think there was a case of polio in the Ukraine at the beginning of the war. Yeah, it disrupts immunization and supplies and so forth. So not good. Mm-hmm. Let's see. Oh, here we go. Will, my brother-in-law caught polio in the late 50s. He walks with braces and crutches. Vaccines were quite rare at that time in China. Well, I was lucky. I was born in 1953, and I wasn't immunized until 55. I was lucky not to contract polio because I might be walking with braces. I know some people my age who have after effects of being paralyzed. Yeah. Yep. A little information. The organization was founded by FDR in 1938 as the National Foundation for Infantile Paralysis to Combat Polio. The name March of Dimes was coined by Eddie Cantor. That's very good. Celebrity medicine. Now we have celebrity anti-medicine, right? Thank you for that, Craig. That's a huge mistake. Now, you're referring to the polio story I told. Were they not detected during clinical trial, these revertants? Okay, so this is... Here's the issue. So Sabin could not do a clinical trial in the U.S. because he was told, we have IPV, we don't need your stinking vaccine. So he went to the Soviet Union, and Chumakov, the father of Kostya Chumakov, he was friends with Sabin, and they said, yeah, we will do big clinical trial for you. Okay. They put it in many, many millions and millions of kids. Totally protective. No reports of vaccine-associated polio. And Dorothy Horstman, a Yale virologist, she went and checked the data. She said it all looks good. So did they not have vaccine-associated polio? I asked this of Koistcha Chumikov in my TWIV with him. He said, when you do massive immunizations, everybody is immune, so you're less likely to see that. And now we don't do massive immunizations. We do a little bit here, a little mop up here. So I think it wasn't detected initially. And then later when we started doing smaller vaccinations, we found it. Yeah. Can you remind us again how many days after COVID illness we should wait until we get a vaccination? I don't remember either. Amy, do you remember what Daniel says? "30 days? Let me look it up. 2 months is according to the CDC. Yeah, 2 months. You should wait after your infection. Okay. 2 questions. Is procorna replication cyclophilin A dependent? I don't believe it is. Dr. Rosenfeld, may you please weigh in? I am not aware that it's cyclophilin A dependent. No. I performed my lab did a screen with Charlie Rice's lab to identify restriction factors. Yeah. Yeah, we found one potent restriction factor that restricted polio replication. I don't remember the name of it. We tried to follow it up. We never got anywhere. And, of course, nobody's working on polio anymore. So we don't know. None of that has been followed up for poliovirus, at least. What's the name of that restriction factor, Amy? I'm asking Amy all these questions. Maybe she's putting the answers way down in the chat. No, I don't see them. Amy, you still there? No neurotropism for SARS-CoV-2. No neuroinvasion either. Thank you very much. I believe what Dr. Rosenfeld is saying here. Now, Amy, tell me, is polio cyclophilin A dependent? And what was the restriction factor that we found with the rice screen? Rice screen. Could a subunit vaccine work for poliovirus? No, I don't think it will work. I think even the full capsid vaccines are not great. Something else is needed. Yeah. Poliovirus recombination with other enteroviruses. This is remarkable and interesting. Could you elaborate? We're full of enteroviruses. They're all over. It's one of the more common viruses you can identify when you do nucleic acid sequencing. We have a lot in our intestines. So the polio vaccine is ingested, starts to reproduce in the intestine, and there the RNA recombines with other enteroviruses. When 2 viruses enter the same cell, their RNA will be copied by the polymerases, and they can change templates while they're copying, so you can get pieces of genome from one virus and the other in one single RNA. And it's very prolific. It happens at high frequency. So when you get OPV, after a certain amount of time, many of the viruses you shed are recombinants with the enteroviruses that are in your gut, like Coxsackie viruses and other enteroviruses. And this is really important. And this is why any entero vaccine, an infectious attenuated entero vaccine that you take orally is probably not going to work, because it'll recombine with these gut enteroviruses. And this is the interesting thing. The Sabin vaccine strains have lost fitness. His process of attenuating them caused a reduction in their fitness. So they reproduce less well, and they're less likely to invade the CNS. But there's very strong selection for return of fitness, right? Restoration of fitness. And that's what happens as these viruses reproduce in the gut. Anything that restores fitness is selected for. And and so if you have a back mutation of a change in OPV, that restores fitness. And NOPV2, to its credit, prevented the back mutation in the 5' non-coding region. But if you have recombination that restores fitness, that is highly selected for as well. And so the rest of the genome, with all the changes introduced into NOPV2, they're gone, and now that virus is fit. It's got some random enterovirus sequence. It doesn't matter. As long as it's fit, it will reproduce to higher titers, and it will enter the central nervous system and cause paralysis in a certain fraction of people. So it's all about selection for fitness. And in fact, OPV itself, most of the virus you excrete after a certain period of time, they're all recombinants. And they've regained a lot of their neurovirulence, but really what they're regaining is their fitness. We assay for neurovirulence, but really it's fitness. I have had a revelation in my later years. It's all about fitness when we talk about viruses changing their pathogenic properties. So the Marek's disease virus that we talked about on TWIV, over the years, it's become more virulent, kills more chickens, but that was not selected for. What was selected for was an increase in fitness, the ability to replicate. And that caused, as a corollary, an increase in virulence. And so do not ever talk about selection for increased virulence, or decreased for that matter, because that's not what it's working on. It's working on fitness. Okay, Tom, hope that goes. mRNA vaccines for polio, I think they've been tried and they're not good. I will let Dr. Rosenfeld comment on that. mRNA vaccines. Do other viruses do this rebound thing? All right, there's no rebound thing. Many viruses have a biphasic disease course. It's not rebound. What the hell? What are we giving these names for? I'm not blaming you, Phase Star. I'm just taking off on your cue and I'm going to rant. Why do we call it a rebound? You know, in the old days, you would study this in animals. Okay, there's a viremic phase with symptoms. The viremia goes away. And then there's an inflammatory phase with other symptoms, different symptoms. Not rebound. Now, the problem is that, yeah, the antigen tests become positive. That's BS. It's just some antigen hanging around. The tests are not great, and it became positive again. It was never negative to begin with. Don't conflate the two. There's no rebound. It's the course of disease. There's a viral phase. Then there's an inflammatory phase. That's the way it works. It works for many viral diseases. People seem to think that SARS-CoV-2 invented everything new in virology. You know what, folks? It's not true. It didn't. A lot of people didn't know how other viruses work. And a lot of people came in the field and didn't even care to learn about them. So they're, oh, look, this is new. Yeah, do you think it's new? Why don't you go read the literature? Oh, my God, you get me started here. DR says, what made the smallpox program so successful? Is that considered a sterilizing vaccination? No, it is not a sterilizing vaccination. You still can get infected after vaccination. You don't develop the rash and the severe disease. What made it so successful? 3 things. Smallpox virus only replicates in one host. It gives you lifelong immunity. And asymptomatic spread does not contribute to epidemiological spread of the virus. So asymptomatically infected people, there are some for smallpox, they don't contribute to spread. So if you see a kid with smallpox, you immunize everybody around that kid, you're going to stop the infection because that's the only person with smallpox virus in that area, the one with the symptoms. There's nobody with a silent infection. That's why it was so successful. Of course, even higher level, D.A. Henderson was an amazing organizational guy who got this whole thing to work. And that's just a great story. And in fact, where is it? I have the book right here. All right, there's a book about smallpox. All right, I've run out of time. I can't find it. By D.A. Henderson himself. And you should try it. Wait, is this it? No. D.A. Henderson wrote a book on smallpox eradication. You should read it. It's really fabulous. So we had some biology and some, organizational stuff. Anonymous entity says mRNA will also be inducing humoral immunity. Yeah, of course. mRNA makes proteins that induce humoral and cellular immunity. Both, yes, for sure. How is SARS-CoV-2 getting around the body without viremia? It doesn't reproduce in neurons. That's a good point, though. There is no viremia. There is no infectious virus in the blood of people who have SARS-CoV-2. PCR is not infectious virus, folks. So, what's going on in the brain? Yeah, there's pieces of RNA in the blood, so they can get in the brain as well. I don't care. It means nothing. And, there's the virus that goes in your olfactory epithelium, and it messes up the sustentacular cells, the support cells from the neurons, but doesn't touch the neurons. And the messing up of the sustentacular cells is why you lose your smell and taste and so forth. But virus does not get in the brain. I've never seen anything convincing that says it gets in the brain. Yeah, you can do it in animals. I don't care. I want to know in people. People are what we're interested in. Animals tell us a lot, but they don't, on many cases, definitively answer things like, does the virus replicate in the human brain? You need to look in humans. Something concerning me: when it rains in Mexico, California, the sewer backs up, spills into the Pacific. Well, yeah, what's going in the Pacific are fecal coliforms and everything. Well, it's feces, right? And coliforms are used to measure. They measure the coliforms like E. coli and related bacteria. And they say, oh, the coliform count is above this, you can't swim. That means there's a lot of poop in the water from the sewers. That's all it means. It's nothing unusual, but in people's intestines, there are many pathogenic bacteria, so you don't want to swim in it. You should thank them for it, because then eventually they go down and you can go back in the water. Okay, Millie, good question. Hey, we have 242 people. We're almost reached the Daniel Griffin number of 300. Now, nobody can match the Amy number, of course, but hit the like button, folks. Get more people to listen to good science. In 1960, polio vaccines got contaminated with SV40. Could it be that the virus is part of the mutant recombinant protein now called COVID-19, and that is why people are shedding it? No, absolutely not. There's no relationship between SV40 and COVID-19. I can assure you. I mean, you don't have to believe me. You can just look at the sequence, but there's no relationship, no. Do viruses actually look like the pictures that often get shown, or are they just representations? Well, it depends on what you're looking at. You have artists who draw viruses, right? And then you have people who 3D print models of viruses. So, does this look like SARS-CoV-2? No, not at all. I mean, generally there's spikes and there's a blob with RNA inside it, but probably doesn't look. If you look at an electron microscope, then you see sort of what the virus looks like. But remember, you're staining it, and it's possible that it's full of artifacts. In fact, the picture of herpes simplex virus that was on the internet for years, which was used to make the giant viruses. No, the plushy viruses. What are they? These things. Right. This is an influenza virus. Giant microbes, right? They made one of herpes, which was wrong. It was based on the EM that had been circulating for years, and it was wrong. It was an artifact of the staining procedure. So the staining procedure itself can cause artifacts. I'm trying to get this to smile at you. There, actually, it has a face. Look at that. Never noticed that. So even the electron micrographs are suspect. Now, of course, you can solve the structure of a virus, and then you can do 3D printing based on the structure. So this is poliovirus, right? You know, the surface probably looks similar. But remember, a virus isn't a static thing like this. This plastic is not moving. All the molecules in a virus are moving very fast. And who knows, you know, that's going to give the surface some kind of plasticity, right? If you could just make yourself small and go look, you'd probably see this thing vibrating like crazy. So everything's an approximation. We do our best. You know, look at this, like this. This is an approximation of a snake, right? We're all approximating. Okay, let's see. These are good questions. First time you've managed to catch a live stream. Yeah, good for you, Jenny. Welcome. I'm particularly animated tonight. Why is it there are only 3 serotypes of polio, where with many other RNA viruses we find many more serotypes? Well, but not all RNA viruses, right? There is one serotype of measles. There is one serotype of mumps and rubella. Why? Well, why questions are tough in biology. But we think for poliovirus, I don't know why there are 3. Why are there not 4? Why 3 and not 2 or 1? I think it's a matter of all right, so I'm going to say the antigenic space. So the viruses are circulating in people. People are making antibodies and T cells, and the virus changes over many, many years to perhaps slightly evade immunity. And 3 serotypes is what results. So you can't make a 4th one. It can't change enough to make a 4th serotype. But I think the capsid constrains the antigenicity, the ability of the virus to change. I mean, certainly polioviruses and enteroviruses do change, right? The genome undergoes mutation at every cycle. There are changes transmitted to the proteins. But yeah, they're just 3 serotypes. Whereas influenza virus, you have new variants every year that are substantially different and require making a new vaccine. We have SARS-CoV-2 variants, but those are envelope viruses. And I think there's more structural plasticity in an envelope virus to allow those changes, whereas we're constrained by the capsid here of poliovirus. But then again, you know, measles virus is enveloped and it's only 1 serotype. So the hell with that theory, Vincent. Yeah, I agree. I don't think we really know. All right, Amy says, no, not antigenic space. Okay, then what is it that constrains 3 serotypes? Put the answer in, Amy. Not antigenic space. Okay, that's fine. I was just throwing stuff out there. All right, Marty says, let's get pedantic. We say, I got the flu. Can we say, I got the COVID? I heard this and I was, well, COVID is a disease, right? I got the COVID disease. You could say that. Daniel says it kind of half jokingly. I got the COVID, because that's what people say. I got the flu, which is a disease. I got the COVID. But what are you going to do? Say I got infected with SARS-CoV-2? People will say, you mean COVID? So it's okay to say I got COVID because you got the disease. I'm okay with that. Hello, Noir. Welcome from New Mexico. What do you think about VZV vaccines as defense against herpes 1-2? They have T-cell cross-reactivity. There was a recent paper showing VZV titers predict symptomaticity. I didn't see that. I think that's a good idea. I I would like to look at that paper and check it out. But if there is sufficient cross-reactivity, it's great. Because we have Shingrix, right, the VZV vaccine, which is really good. Yep. I think it's a good idea. I like it. Okay, John says, thank you for your contribution to science communication, by the way. We really appreciate it. In the spirit of beginning a semester, what does obligate mean with regard to the definition of a virus? Obligate means it has to happen. So we call viruses obligate intracellular parasites. So we don't need the parasite part. Obligate intracellular means they have to get inside of a cell in order to reproduce. In order to make more viruses, they need to get inside. They will not reproduce outside of a cell like bacteria. Many bacteria will reproduce on their own outside of a cell, in broth or in your blood or in your intestine. But viruses cannot do that. They are obligate. They have to get into cells. So that's a good question. I like that very much. Thank you. Oh, this is a good question. And Dr. Rosenfeld may know. Vaccination against poliovirus can use intradermal way like smallpox. So it was tried. Fractional intradermal IPV was tested by the group at the University of Vermont, where I did a TWIV not too long ago, and it was suboptimal in terms of antigenicity. So not happening. Not happening. Will, sorry for the basic question. First of all, never apologize for any question of any simplicity or complexity. You can ask me, what's your name? I have no problem with that. All right? Even if you ask it every week and you have been here every week, it's fine. You can ask me my name. So any question goes. All right, what antibodies are mentioned? Do these prevent disease, or is that dependent on T cells? Okay, so antibodies will bind. Here's a spike protein of SARS-CoV-2. So the antibody will latch onto the spike and prevent the virus from infecting a cell. So it will prevent infection of cells. Now, the problem is when let's say you're vaccinated, you've been vaccinated 6 months ago against SARS-CoV-2, you get infected, the virus comes in, there are no antibodies there. The virus starts to reproduce, and then your body says, SARS-CoV-2 is here, let's make antibodies. So they recruit some memory cells that know how to make antibodies. And then a few days after you're infected, you start to make antibodies. And depending on the strength of your response, that could reduce viral titers enough to really impact disease. But everybody's different. Everybody responds differently in terms of immune responses. So in another person, it may start reproducing a lot, in which case then T cells are there. They will kill virus-infected cells. So I think it's, we have this dogma which says T cells will prevent severe disease, and they certainly will clear virus-infected cells. But I think the two of them make a good combination together, and that's why we have them both, right? Because they're both useful together. Is there any research being done on trying to prevent the myelitis rather than infection? Well, we're not trying to prevent infection. So anyway, there is none. There's no research on that. What we're trying to do is, and not even prevent infection. We're trying to reduce virus titers in the blood so it doesn't get into the brain and cause paralysis. That's the goal of the vaccines, to prevent paralytic disease. So that's the easiest target. I think preventing myelitis would be very hard, because what are you going to do? Let's say you had therapy for preventing myelitis. I don't know what that would be. So you have a kid infected with polio. You're going to give him the therapy right away? Or are you going to wait until they get paralyzed, and then it's probably too late? So you see there are all kinds of timing issues involved there. Hmm. A lot of good questions here. Poliovirus. You're suffused with energy. It becomes you. Thank you very much. I usually have a lot of energy, but the questions have been particularly interesting. Yeah, they're always interesting. I don't know. I can't explain it. Maybe I had a good dinner. I don't know. Maybe it's because there are 246 people here. I don't know. Didn't Joni Mitchell have polio? I don't know. Did she? That doesn't ring a bell. Let's search it. She contracted polio at age 9 and was hospitalized for weeks. And Song for Sharon supposedly talks about him. So yeah, you're right. She did have polio. How about that? We need to teach information literacy. In other words, how to distinguish between good information and fake information or false information. I think that's really hard, right? You kind of need to know a field. But I don't know, how would you do that? I mean, you do a Google search and you get all these results. How do you distinguish between what's right or wrong, especially with technical subjects? So I agree with you. We need to, but I don't know how you would do that. GSD Bio didn't explore microbe TV until 2023. Now I'm a big fan. Spread the word. Spread the world. Sorry, spread the word. Please, please do. Lise says, I tell the Ohio State researchers all the time about microbe TV. I pass links to them. They pass the info on to others. I mean, you folks here, 250 of you, you're crucial because you're here every week. You listen to the pods. You listen to what me and my guests are saying, and you spread the word. And sometimes you get other people to listen. So just keep doing that. We're slowly moving. I mean, we're going to get to 127,000 YouTube subscribers soon. We get about 800 a month. We need to increase that because we want to reach more people. But we will. I am confident. How did the monkeys get SV40? Well, all the monkeys in the wild are all infected with SV40. And we used to take kidneys out of them, capture them, take kidneys, and use it to grow poliovirus. So, in the 50s, we had no clue that there were viruses in the monkeys used for that. We had no clue. We don't do that anymore. Would love to hear polio-related stand-up. I'm not sure. I think that's iffy, right? Because it's a paralysis, and I don't think we should make jokes about it. But other things, here Visto says, OPV2 and OPV3 walk into a bar, and then what happens? You gotta segue, right? China news. I check China CDC every day. Will lives in Changsha. There's lots of influenza A and B now, RSV, mycoplasma, virtually nothing about COVID. In December, they stated COVID was at its lowest level for 12 months. Thank you. Even exponential spread of microbe TV takes time to make a big impression. Nobody in the general public thought much about SARS-CoV-2 for the first few months. Probably, yeah. When I have low fever, 37.3, after surgical mask wearing at the pharyngeal site, temperature is 39, and maybe that temperature stops virus replication. I don't think so. All right, Amy says Yitzhak Perlman also had polio. That's correct. And no stand-up for polio. She said, no, we don't want to do polio stand-up. Okay. Thank you for being with us tonight, Dr. Rosenfeld. I don't know that, Dr. Rosenfeld, what do you think about 39 degrees inhibiting virus replication? Fever. What do you think about that? I was always skeptical. Mm. Especially elevated cytokine. Histamine is notorious to incite anxiety of crossing the blood-brain barrier to the brain. Yes. Still masking to avoid long COVID and long-term sequelae. I don't understand why there is so little discussion of the long-term neurological and cardiovascular risks. Well, it depends where you're listening. In some circles, there is a lot of discussion. Daniel Griffin talks about it every week. And there's certainly long-term neurological and cardiovascular risks of after infection. And people are trying to sort out their etiology, how they happen, and maybe then you figure out how to prevent them, right? It's not going to happen overnight. It's not going to happen in 10 years. It's going to take a long time. I mean, I know that's not what people want to hear. But most people are not masking because this is not in their head, and most people do not have long COVID. They're taking a risk. They're doing a risk-benefit. I want to have a life without a mask, and frankly, that's their choice, right? If I tried to tell my old ex-doctor, that was your old ex-doctor or your old ex-doctor? I guess it's your old ex-doctor. To listen to this channel, they would tell me I can't take information from the internet and shut it down. Yeah. But Daniel Griffin is not just the internet, right? Daniel Griffin is a doctor who uses the internet to spread good information. So I don't think you can blanket say I'm not going to use information from the internet. There's a lot of good information out there. Many doctors will listen. If you're a patient, you go in and you've done some research, they'll listen to you. Not all will, I agree. But many doctors will listen to you and say, yeah, that's very interesting, and here's why it applies to you, or here's why it doesn't apply to you. And I do appreciate that very much. So here we go. I tested positive for Thursday evening. By Friday afternoon, I started Paxlovid. What a lifesaver. Makes you feel much better. Yep. Hello, Tona. Oh, Rio Grande. Wonderful. Puerto Rico. Enjoy the warm weather. See, Nick Zentner's Washington Geology Channel. He's revolutionizing teaching like you. He takes the world on field trips, and we even help research with Google Earth and historic data finding. That's great. Sounds like he's doing really cool stuff. I wish I could do more, frankly. I wish I could do field trips. I always wanted to do that, like a cruise. Hey, a Hudson River cruise. One day up and down the Hudson, and I get a couple of virologists. We give lectures. We mingle with you guys at meals. I think that would be very cool. It's another way of teaching. But I don't have the capacity to organize that right now. I can't do it. Maybe if the company gets bigger, we can do it. I just don't know. Right now, we're trying to spread the word, right? Could drugs like Paxlovid drive mutation? All right, so sure, antivirals, antibacterials, they all can drive. They can select. They don't drive mutation, okay? They select for variants that are resistant. The mutation occurs in the reproduction of the virus or the bacterium, and then if there's a mutation in the population that confers resistance, it'll be selected for because it'll multiply where the other viruses don't, right? So in theory, yes, you could select for Paxlovid resistance, but we haven't seen it. And many people have gotten Paxlovid. You can do it in the lab, for sure, but we don't see it in people. What does that mean? Good exam question. Here's the question. You treating X millions of people with Paxlovid, and you fail to see resistance to the drug in circulating viruses. Yet in the laboratory, you can make resistant viruses. Provide an explanation. I'd love to say provide. No, that's too open-ended. They'll take advantage of you. So what is a good answer? The mutation decreases fitness, so it can no longer compete in the wild with other viruses that are, pardon me, cannot I don't have a cold. I just have a tickle in my throat. It cannot compete in the wild with viruses that retain susceptibility to Paxlovid. So it's all about fitness. Where in the laboratory, there's no competition. You're just growing it on its own. So you wouldn't know if it lost fitness. Although you could do that experiment. And that would be part 2 of the question. Design an experiment to determine if resistance to Paxlovid reduces fitness of viruses. You know, I have to tell you folks, you 254 lovely individuals, I never thought 5 years ago that I could sit here in front of a camera live and talk about viruses to my content. I never thought that. And now I have so much fun doing it. You have no idea. I just love it. Thank you very much for making it possible. And here we go. Amy, no neurotropism for SARS-CoV-2. My doc wouldn't give me Paxlovid because it was day 6. Yeah, you need to have it earlier. He or she was right about that. Yep. Amy Rosenfeld comments at 556. Okay, that's a good timestamp to see that. I know a couple of people who have not recovered taste or smell 2 to 3 years after COVID. Yeah. How is that not neurotropic? Dean, I just explained it to you. I would never say that to a student. Okay. The virus is infecting cells in the in the olfactory epithelium, right, which is up there. They're support cells. They're sustentacular cells, not the neurons themselves. The sustentacular cells are needed for the health of the neuron. The virus infects the sustentacular cells. It makes the neurons not function properly. So you don't have to... Why do you think the virus has to be neurotropic to affect a function like that? Because there are other cells around that, for example, well, never mind. That's a bad example. So it's not infecting neurons. It's infecting the sustentacular cells that are required for the olfactory sensoring. Ooh, getting a lot of buzzes on Instagram. What's going on? Will, azuvidine is a repurposed HIV drug. Maybe it's cheaper than Paxlovid. Couldn't it be sold more widely? Well, it's a reverse transcriptase inhibitor, which means it could also inhibit other nucleic acid polymerases. It has conditional approval in China, right, for COVID-19. Yeah, it's probably cheaper because it's been around a while. Couldn't it be sold more widely? Yes, it could be. It has to be, in the U.S., would have to be given a clinical trial. So the company who makes it and who owns the patent would have to do a clinical trial in the U.S., which is enormously expensive, and they may not want to do that. So you may think that science is the ultimate answer for questions like this, but it's not. It's also economics, right? And so you have a drug that works very well. Very hard to get another one in unless you're willing to spend a lot of money, and many companies not willing to do that. We have a question here. So, neurotropic means the virus must be replicating in the brain. Rabies. As far as I know, rabies virus is replicating in the brain. Amy Rosenfeld, what do you think? Does rabies virus reproduce in the brain? I thought so. As a second grader, I was disappointed they discontinued the polio sugar cube. I remember getting that in 1962 or something like that. A little paper cup, the cube in there with the red color. That's the virus-containing solution, and you just pop it in your mouth. Yeah, it was sweet. Now they've just put drops in your mouth. No more sugar. Yeah. Oh, this is very funny. You should make Amy's name show up in a special color. Well, we could make her... Does the moderator get a special color? Yeah, my moderator is blue. Amy, you want to be a moderator? Then we'll make you a special color. Let me know if you want. Thank you for that suggestion. That's very funny. And here's when Amy appeared, by the way. Hello. But this is a long time ago. I know you've been here for a while. Okay. Not cyclophilin A. So polio is not cyclophilin A dependent. And no subunit, no virus-like particle vaccines for poliovirus. I thought Amy was talking into Microbe TV's ear. Why not? She's always right, or mostly right. We should have an Amy chat box on screen so she can real-time correct Vincent. Now, Visto, why do you think I need to be corrected? Do you think I need to be corrected? Yeah, sometimes I do. That's true. Hey, ApoL6, there it is. That was the polio restriction factor, ApoL6. Highly restricted polio infection, and we wanted to study that, but we never did. And so, yes, Sabin hypothesized that if polio replicated in a different animal outside of humans, it would be attenuated in humans. So he passaged polioviruses in different animals to try and make his vaccines. Yeah. Well, I'm not going to highlight this comment, but there's a comment here about... there's a YouTube channel that says that DNA in vaccines causes cancer, which, by the way, no one has ever shown and is highly unlikely. So don't believe any of it. You know, first they said the mRNA vaccine was a problem. Now, of course, people now realize, because they don't think about this, all vaccines have DNA in them. So, now all vaccines cause cancer. Really? This is so ignorant. It's unbelievable. Anyway, so this person goes on this other person's channel who is saying that DNA vaccines cause cancer, and his comments, and he's trying to debunk it, and his comments get deleted. So you go on a channel that's spewing nonsense. You put the truth there, and they delete you. Someone asked me before if I believe in free speech. Obviously, they don't, because they're deleting comments that are counter to what they think, which is wrong. Now, I admit I do delete comments that are offensive. I delete comments that say, my doctor cured me of herpes, because that's not possible. You either didn't have it or you're lying, because there's no cure for herpes, okay? Permanent. Yeah, if you have a cold sore, you can put some of that acyclovir cream on it. It's not curing it. The virus is still in you. Fitness of CVDPV is not known. That's correct. Nobody studies it because they're shutting down all the polio labs. And one of the least intelligent actions ever to happen in the science world, polio research is being shut down. Why? Why? The world is full of poliovirus. Why can't we work on it? It's in your sewers. As I said, people haven't thought about it. No mRNA vaccines for polio. There you go. Does increased fitness go hand in hand with augmented interferon antagonism? Could. I don't see why not. Immune antagonism could be a reason for increased fitness, yes. Thanks, Amy, for stopping by. Jamie, hello. Jamie visited the incubator last week. The doorman is still talking about your visit. The doorman in my building said, "Oh, that guy Jamie, he was so excited to come visit you. What are you doing up there anyway?" It was so funny, Jamie. Anyway, thank you for stopping by. And yeah, he's thanking Amy. Blaze says, print a picture of Amy and tape it to your screen. Oh, I have this thing here. Did you see the Amy virologist? I mean, I could leave it there just to signify that she's here. Right? When Amy is in the house, we put that up. How's that? Abdul, thank you so much for your contribution. Is it unethical to give OPV after IPV? I think not, because once you have a dose of IPV, then you will not get paralysis from OPV. So I think that would be good. If you did that worldwide, that would be good. But then you're giving a shot of IPV to everybody. That's going to be hard. But I think you've solved the problem, yeah. Okay, you want me to leave the plaque assay princess? All right, I'll leave it up there. Yeah, I am. I put up the plaque assay princess. There she is. When there is a huge problem with something resembling leptospirosis in Ukrainian trenches. I have not. You have to point me to a source where I can investigate it, and I think that would be something for TWIM this week in microbiology. Smallpox shedding after pox formation. Yeah, okay, that happens. That's how it's transmitted, right? But there are no asymptomatic infections, right? You agree with that? Oh, this is a good point. Did Vincent talk about the measles outbreak in Philly? I did not, but we'll cover it on Friday on TWIV, right? What a great group. What a great group. I love it. Severe disease of SARS-CoV-2 is caused by aberrant cytokines and immune response. Age, if you're old, that does it, and also antibodies to interferon, yeah. Look at this, Amy. I feel if Amy came back occasionally, attendance would increase skyrocket. How you doing, Amy? You like that comment? Make you feel good, right? Amy was my YouTube highlight of the week. I'm trying to make Amy feel good. Casey says, wasn't there a paper where they recovered replication competent SARS-CoV-2 from the brains of COVID-dead patients? There have been a mix, right, where they don't find it, and then a few where they do. In this case, they didn't quantify it. They just said there's infectious virus there, but you don't know if that was actually replicating, and you don't know if it contributed to disease. So it's problematic, yeah. No. What's the no for, Amy? I don't know. It came so far after I said it. You want me to turn off your picture? Send me a text if you want me to turn off your picture. Okay, I got my message thing open. Just do it if you want me to turn it off. Maybe you don't like it up. Quiero uno de esos. What is that? I don't know what that means. Let me write it in a translate program. Quiero. I'm sure someone could do this faster than me. Uno de esos. I want one of those. What do you want? What is it that you saw? Let me do it in Spanish, huh? What do you want? ¿Qué decías? ¿Qué decías? I bet it was the shirt, right? Yeah. I bet it was the shirt. Structural. It's structural. The limitation of serotypes is structural. So notice Amy is very efficient with words, right? Structural constraints. One word per post. Thank you, Amy. Thank you. I know, Casey, that's what they wrote, but it's wrong. It's wrong. Sorry, we did that paper on TWIV. Not a proper conclusion. Not a proper conclusion. Can you explain what happens when someone gets COVID-19, tests Paxlovid, then tests okay, I can explain what happens. The negative test isn't right, okay, because, you swab your nose and maybe you missed where the virus was. Maybe there was a little bit of virus stashed away somewhere and it came out 3 days later. So, a single negative followed by a positive, I don't think you were really negative to begin with. I think you're shedding antigen. I don't think you're infectious any longer after 5-6 days. And the antigen tests at that point are iffy. That's what I think is happening. Structural constraints on the particle size also. Yes, we're talking about poliovirus and 3 serotypes. And Ian says, what is the frequency of errors in replication for viruses? Is there proofreading? It's amazing that mammals have viable offspring the majority of the time. So, the error rate for us, for mammals, is about 1 mistake per million bases. And for DNA viruses, it's similar. But for RNA viruses, it's 1 mistake per 10,000 bases, right? Those are the error frequencies of the polymerase. Many of those errors get into viruses. Some of them are lethal, so you never see those viruses. Some of them are neutral, have no effect, and then some of them confer some fitness advantage at some point, like drug resistance or temperature resistance or something else. But we need to have mutation frequencies to survive. We cannot evolve without mutation. So you can take E. coli and make it mutate less, and it is not competitive. It's not fit. And so the same is with mammals. We need a certain amount of mutation. We can correct a lot of the errors. So you ask about proofreading. DNA viruses and us, we have proofreading mechanisms. So DNA polymerases have proofreaders associated with them. Only one group of viruses, RNA viruses, have proofreading, and that's the coronaviruses. All the other RNA viruses do not have proofreading function, and that's why their error rate is higher. Stein et al. Isolated replication competent virus. Does that suggest it's neurotropic? No, it doesn't. You have to show the virus is in a neural cell replicating, because you don't know how that particular virus got there. You don't know if there was one particle or many. It may have just come through the blood, flowing through on its way. So, no, it's not enough. No. I mean, I agree that we should know, but I haven't seen any experiment that's definitive. Here's an interesting idea. It's illegal to holler fire in a theater when there is no fire. So it should be illegal to badmouth vaccines on YouTube. That lie is killing people. Yeah, there's a lot of things that should be illegal. But I do agree with that sentiment, John. Intradermal IPV, no. Someone asked about that. Nope. Here's a good question. Well, they're all good. Has there been a study to sequence the genomes of individuals who have been paralyzed from polio to see if there are variants of genomics or genomic signatures in common? Great question. There is one study I'm aware of from Denmark, and they looked for these genome-wide association studies for variants in people who had polio. And in fact, I traveled to Denmark a number of years ago, and I interviewed the person who did that work, so you could search for it on TWIV. They didn't find anything convincing. We need more numbers of people, but no one seems to be doing it. No. But yeah, that would be an interesting thing to know, if there is some genetic predisposition to getting paralysis. Yeah. I think they do have a immune defect of some sort. So people who get paralyzed, kids who get VAP, I think they have an immune defect which allows virus to reproduce to higher levels and thereby invade the central nervous system, right? Ian says, I described vaccination as higher education for the immune system. I think it's education. I don't think it needs to be higher. I like it. It's a good one. It's really good. Vaccines prevent neuroinvasion. That's right. Polio vaccines reduce virus load in the blood, so it's less likely that the virus is going to cross into the brain. No crossing the blood-brain barrier. Polio virus itself that does the damage in nerves, or is it immune collateral damage? Very good question, which we tried to study in my lab. No answer. We don't know. But Amy thinks the majority of polio deaths are by virus killing neurons. Okay. I think it's kind of up in the air. Also, Amy says those who survived polio paralysis was transient and due to the immune response. So it's both. I read an article this morning about intranasal vaccine development using mastopiran. It's a peptide toxin from wasp venom, and it's a adjuvant. I don't know how it's an adjuvant. I'd have to look into it, but it is from wasp toxin. So you can imagine if this is ever used in a product, the anti-vaxxers are going to go crazy. How do they make crystals of giant molecules like DNA and proteins? Fills me with wonder when I look at those diagrams. It's not easy. It's hit or miss. And nowadays, we do structural determinations that don't require crystals anymore. We just go cryo-EM, where you can just freeze the molecules and solve their structures. Crystallization is very difficult. Yeah. Judy Collins also had polio. Oh, so now tonight we have a range of celebrities who had polio. We have Judy Collins, we had Yitzhak Perlman, we had Neil Young, and Joni Mitchell. And, of course, FDR, right? Many more, of course. Is there any evidence that OPV has better non-targeted effects? Not sure what you mean by that. Sorry. When I took my dog to the dog park, I would share your discussions with others in TWIV. Now I avoid the dog park on my veterinarian's advice because of the pathogens making dogs so sick. Yeah, dog parks are a problem. Yeah, Amy, I don't know what are non-targeted effects when talking about OPV. Sorry about that. All right. Let's see. We have a lot of questions left, but we don't have time to cover them. The Hudson River cruise would be great. Let me research it. Please, if folks would like to help, I would love to do that. And I could get people to go on the cruise. I'd go myself, but I can't do the logistics. Okay. Everyone now is happy that I'm happy to talk about viruses. All right. I'm in a great mood. It's way past my birthday. I don't know. Sometimes I'm usually upbeat on these things. It's just sometimes more upbeat than others, right? Hello. Talia 7 from the Philippines. First Philippines tonight. Amy says rabies replicates in the neurons of the brain. There you go. Definitely give Amy a wrench. I'll be happy to make you a mod. And then you'll be blue. For sure. If a virus suddenly grew to the size of a human and wanted to kill you, how would you kill it? A gun, inject it with a giant dose of Paxlovid, run. Well, if you could break it, yeah, you could break it. All you have to do is break it, a big hammer or a gun. Both Paxlovid, no good because it's only going to hit replicating viruses, and it'll run faster than you, so you can't run away from it. pass a link to this show to Dr. Campbell. I think many people have tried, and it doesn't work. Oh, okay. Amy doesn't want to be a moderator. All right. Thank you, Amy. We can't highlight her in a different color. But when you come, we will put your picture up here out of acknowledgement. All right. That is 10 o'clock. It is time. Now it's time to say goodbye to you and all your friends. Is that how it went? I never left. No. Let's see if there are any more contributions here. Markle says, Amy, we're very fortunate to have met you on Twitter. Thank you for contributing what you can when you can. Oh, here we go. Thank you, SRR, for your contribution to science communication. Yes, your plaque assay princess by Barb Mack, our moderator, who's not here tonight, but she had it made for Amy. Yep. All right. Amy says, autophagy alterations may regulate fitness and ultimately neuroinvasion. Yeah, trying to figure out what regulates fitness of viruses. Many people do not study it. Here's one article on pathogens from the Ukrainian trenches. So there you go. Thank you, Vanity, for that reference. Alan Alda and Robert Redford. What about them? Did they have polio also? My 10-year-old son wants to know if animals can get COVID. They can get infected with SARS-CoV-2 for sure. You know, deer, big time infected, mink, all kinds of wild animals in zoos. Yeah, infects almost everything probably. Amazing. All right, folks, I think that does it for tonight. Thanks all for coming. Really nice. Thanks, Amy, for coming. I want to thank the moderators tonight. We had, so Robert Redford and Alan Alda both had polio. I want to thank the mods for tonight. We had Les, we had Steph, we had Tom, we had Andrew, we had Les, Steph, Tom, Andrew. Frank was here. Who am I missing? Vanity Nutrition. There you go. Thank you all for moderating. I really appreciate your coming pretty consistently over the weeks. We will be back next week. Our guest will be Cindy Leifer from Immune. So bring your immunology questions next week. So if you want to know the date, it's Wednesday, the 17th of January. January's zipping by. It's the first day of my class, too. But I should be able to make it here for the live stream. I did it last year. I can do it this year. Cindy Leifer will be here. Bring your immunology questions, any kind of immunology questions, whatever you want to bring. And meanwhile, folks, thank you all for coming. You guys are great. You have great questions. I love interacting with you. You provoke me. You make me better. So thank you all for that. And until next week, please take care and good night. Bye-bye.